Okay, so I think we're ready to get started. And so first, just to say hello, my name is Kelly and I'll be hosting the Journal Club today. We have with us today Araya Eukubay, who is a PhD student, currently completing his PhD at Addis Ababa University. And he is also an associate researcher at the Ethiopian Public Health Institute. And his primary interests are looking at insecticide resistance, monitoring vector populations, and looking at ways to employ vector control methods. And so it just leaves me then to pass over to Araya. And Araya, if you're ready, if you wouldn't mind starting your presentation.
Okay, thank you, Dr. Kelly, for the nice introduction. And good afternoon and good afternoon and good morning, everyone, wherever you are, depending on your location. So I'm happy today to be here to share our recent finding. And let me share my screen. Okay, I think everyone can see my slide.
Yes, we can see them.
Okay. So I will be talking on spatial and temporal signatures of genomic insecticide resistance in the Anopheles arabiensis mosquito malaria vector from Ethiopia. And in my presentation, I will just follow a brief introduction, why this study matter and how we approach and what are the key findings and their implication for malaria vector control. So to begin with, we know that despite the huge investment for malaria control, the disease continues to rise and in burden. As we all know, by 2025, according to the WHO report, there were an estimated of 282 million malaria cases globally. And we know that most of the cases, about 94 malaria cases, are reported from the sub-Saharan African country. And Ethiopia is also one of the sub-Saharan country which is highly burdened with malaria, which reported 4.4 million, even with this recent report. So when we look the burden of malaria in Ethiopia, majority of the landmass, about 75% of the landmass is malarious, where 69% of the populations live in malaria risk area. And also malaria is seasonal, in Ethiopia, the malaria transmission season, there are two major peaks, especially one major peak which occur September to December, and also April to June, another minor peak transmission. So malaria is, seasonal, as well as it is also heterogeneous in the country, where, as you can see over the map, the western lowland of the countries are highly burdened with malaria than that of the other districts or than that of the other regions. So generally, malaria is heterogeneous in Ethiopia. And when we look at the trend across different years, like the global trend, by 2000 and 2005, there was a declining situation in malaria. But with that gaining, Ethiopia has launched malaria elimination strategy starting the 2017. But after 2019, malaria started to upsurge, especially by 2024. There was a huge resurgence in malaria, where greater than 7.3 million malaria cases reported in the country. And this malaria burden is assumed to be contributed by different factors. So conflict and instability, climate change are one of the factors that are assumed to drive that. And one of the other factors is insecticide resistance, which limits the efficacy of vector control. That is also assumed to contribute for this burden of malaria. When we look at the major vector in Ethiopia, Anopheles arabiensis is the primary vector, where it contributes for most of malaria transmission. And we know this vector is also geographically widespread in all malaria endemic regions of the country. It exploits, wide range of habitats, including the lowland areas, the irrigated areas, the highland areas. So we know that vector is opportunistic with anthropophilic biting behavior. So as like that of other sub-Saharan countries, Ethiopia also used the primary vector control tool, which depends on insecticide, that is the long-lasting insecticidal nets and indoor residual spraying. So the long-lasting insecticidal nets were, this has been used for a decade, and pyrethroid-only nets were, scaled up since 2005, and since 2025 now, dual AI nets are being introduced to the country. When we look at IRS was also, implemented since the early 1960s, and a number of insecticides have been changed from time to time because of insecticide resistance. DDT was used at first, then it was replaced by deltamethrin, then propoxer and biondocar, then the primiface methyl, that is organophosphate. Then now the country is using the clothianidin-based insecticides for indoor residual spraying. So this longer time, depending on insecticide for the vector control, is now threatening the efficacy of the major vector control strategies. So as you can see over the map, the major malaria vector in Ethiopia has developed resistance almost to all classes of insecticides which are used for indoor residual spraying and also for the impregnation of the nets. So here we know that resistance is caused by target site mutation and by metabolic detoxification enzymes. However, we don't have evidence what causes the detoxifying enzymes to be increased, or which gene is contributing to overexpress these detoxifying enzymes, and also which genes are under recent selection, and what looks like their seasonal dynamics between season and also across geography. We lack this evidence from Ethiopia, especially for the Anopheles arabiensis vector. When we look at the resistance monitoring strategy, Ethiopia relied on phenotype bioassay using the WHO tube and CDC bottle assay, and also using PCR for limited number of markers. We know this strategy can tell us, whether the vector is killed by a particular insecticide or not, but it can't give us the full picture of insecticide resistant mechanism. So this has to be, complemented with a comprehensive genomic study. So we know recently the genomic surveillance strategy is advancing, and it is uncovering the secret mechanism of insecticide resistance. It enables us to rapid monitoring of known markers, as well as to discover the novel variants which are under recent selection. So to address these gaps in insecticide resistance monitoring, and also to gain insights into the mechanism and the spatial temporal dynamics of insecticide resistance, we explored whole genome sequence of Anopheles arabiensis from Ethiopia across multiple sites. So our objective is we want to investigate the distribution and frequency of both target site and metabolic resistance markers. We also need to look into the novel and known genes which are under recent signal of selection. So our approach, so we sampled Anopheles mosquitoes from different regions of the country across multiple sites, which we assumed these sites can represent the country's heterogeneous eco-epidemiology. And also we sampled these mosquitoes during the major transmission season and also during the minor transmission season. So we used CDC light trap and Propopack aspiration for collection of the Anopheles mosquitoes. And most of our samples were collected during 2023. And we have also additional collection, particularly from central Ethiopia, collected during 2021. So in general, 1,272 whole genome sequence of Anopheles arabiensis was included in this analysis, and the whole genome sequence was conducted as part of MalariaGEN Vector Observatory, and all bioinformatics and data analysis procedures were done following the Anopheles Gambia 1000 genome project. So the key analysis we have done here includes we computed the frequency of non-synonymous SNPs at the target site resistance genes. We also computed the frequency of copy number variation at metabolic resistance genes. And we also performed genome-wide selection scan to look into the novel and known genes which are under recent selection. And we also compare the frequencies that we observed, whether they are significantly different across geography and region. Looking at the key finding, so when we look at the target site resistance markers, we calculated the non-synonymous mutations at the known genes, the VGSS gene, the RDL gene, and the acetylcholinesterase gene. So the VGSS gene is the target site for organochlorine and pyrethroid, and we computed the frequency of non-synonymous mutations, and the findings showed that in the northern Ethiopia, the L995F mutation at the VGS gene is found in high frequency, which reaches from 38 to 94 frequency. We also found lower but appreciable frequencies in western Ethiopia and southern, as well as in the south central part of the northern Ethiopia. But frequencies in other cohorts or in other locations, including the central Rift Valley, were low. When we look at the RDL and acetylcholinesterase genes, we did not find the known SNPs that confer resistance to dihydrin and also to acetylcholine, to the organophosphate at the acetylcholinesterase gene at greater than 5%. Of course, we detected the presence of the A296S, which is a non-synonymous mutation which confer resistance to dihydrin, but the frequency was very low. However, importantly, we did not find the G280S mutation or SNP, which confer resistance to organophosphate at the acetylcholinesterase gene. We know that there are reports of insecticide resistance phenotype against organophosphate in Ethiopia and Anopheles arabiensis. However, we did not detect the target site mutation for the organophosphate as acetylcholinesterase. So this can tell us, the organophosphate resistance in Ethiopia might be driven by the other metabolic resistance conferring genes. So we also tried to compare whether the frequency of VGSC SNPs are significantly different across different regions as well as between the seasons. So we used the chi-square contingency test just to see their significance, and we found the VGSC L995F is significantly higher in the northern Ethiopia, while comparison among other cohorts was not significant. When we look at the seasonal variation, we observed location-specific variations. For example, in most cases, we observed the L995F was increased during major malaria transmission season, example in western Ethiopia and southern Ethiopia. However, we also observed in other locations that the L995F frequency was increased even during minor malaria transmission season. So this was, a location-specific variation. Since we don't have seasonal data across multiple years, it is difficult to compare their consistency. However, with the data we have, we detected variation across season as well, but these variations are location specific. So generally, our findings showed that the VGS target site resistance variants are heterogeneous across geography as well as across season, despite the similar interventions that the country has been implementing. Looking at the metabolic resistance markers, so we also computed the frequency of copy number variation at the known metabolic resistance conferring genes targeting the CYP450 and the carboxylesterase gene, as well as the GST2 gene. And our findings showed that at the CYP450 gene, we detected high frequency of copy number variation, particularly for the CYP6AAP gene cluster in western Ethiopia, followed by one site from northern Ethiopia, which we call is a location is Wakamba, and also in southern Ethiopia, while frequencies in the central Great Rift Valley of Ethiopia and southwestern were very much low. In addition, we also found CYP9K1 copy number variation was consistently higher across all cohorts, but the frequencies were much higher in the central Rift Valley than the western and the southern part of Ethiopia. We also look into the copy number variation as the carboxyl esterase gene, and we found the carboxyl esterase gene cluster, particularly carboxyl esterase 2 to 7 Gs frequency, were found at appreciable frequencies in the western and northernmost Ethiopia, while in the other locations, frequencies of copy number variation were very much low. When we look at the GSTE2 gene, the copy number variation frequency of GSTE2 gene was consistently higher again across all locations, but it was very much low in western and also the northern part of Ethiopia, where we found high frequency of CYP6AFP gene cluster. So, but in the central Great Rift Valley of Ethiopia and also southern part of Ethiopia, we have detected high frequency, even near to fixation of the glutathione S transferase 2 gene. We also tried to analyze the SNPs at the GSTE2 gene to detect whether the known SNPs that confer resistance to organophosphate and pyrethroid, the known SNP L99, L119V and I114T SNPs, but importantly, we did not find these SNPs in the Ethiopian Anopheles arabiensis population. Of course, we have detected other SNPs, but at higher frequency, but these SNPs are not yet confirmed whether they do have an impact on the insecticide resistance phenotypes or not. This this will be validated for next. So we also compared the frequencies across the different geographic areas, and like that of what we did for the target site resistance frequency. So the CYP6AFP gene copy number variation were significantly higher in the western Ethiopia compared to other locations. And also the CYP9K1 was statistically higher in the central Great Rift Valley compared to others, while comparison among other locations were not significant. And GSTE2 gene was significantly higher in most sites, but it was significantly lower in western Ethiopia, where we found high frequency of the CYP6AFP gene cluster. When we look at the seasonal variation, we also observed location-specific variation. In most cases, we observed the CYP6AFP gene and CYP9K1 copy number variation increased during major malaria transmission season, but these were also location-specific. In others, we also observed frequencies increasing during the minor malaria transmission season. So our finding indicated that distinct metabolic pathways are driving resistance in different regions of Ethiopia. So resistance conferring genes are very much heterogeneous across geography as well as across season. We also performed the genome-wide selection scan to look into the known and novel markers which are under recent selection. And when we we performed this for chromosome 2 and chromosome 3 arms. And when we look at the chromosome 2 arm, we detected a novel signal of selection, which centered around the 22 million base pair, and it is centered on the genes carboxyl esterase juvenile hormone gene cluster. And these genes are, their primary function is, for regulation of hormone, but recent evidence shows that members of this gene cluster are also involving in metabolic insecticide resistance. So this gene was detected only in the northern Ethiopia, and we don't have evidence that directly link the insecticide phenotype relationship of this gene in Ethiopia, but we found it under recent selection, so this needs a further validation. We assume it could be also involved in driving resistance. We also observed signal of selection near the VGS gene in that northern part of Ethiopia, in line with the L995 higher frequency. Additionally, we also found another novel signal of selection which centered in detoxification pathway, such as the ubiquitin proteasome system on 3L chromosome, which centered around the 20 million base pair of the 3L chromosome. We detected this in the western Ethiopia cohorts only from one site, Asosa site, but we did not detect in the other site for this signal of selection. We also look into the 2R chromosome, and we found a pronounced signal of selection centered at the CYP6AAP gene cluster. And we found this signal of selection in all Anopheles arabiensis cohorts from Ethiopia, even in the area where we did not get high frequency of CYP6AAP gene. So this can tell us maybe selection for the CYP6AAP gene cluster might be also driven with other structural variants like SNPs and deletion and inversions. We also tried to see the chromosome 3 arm, and we found signal of selection centered on the GST2 gene, especially on central Great Rift Valley of Ethiopia and on the south central part of northern Ethiopia, that is the Amhara Harbo site. But we did not find the signal of selection or are centered on GST2 gene from western and northernmost Ethiopia, where we from the northern and western part of Ethiopia, we did not see any signal of selection, but we found signal of selection from the central Great Rift Valley of Ethiopia, where we found even high frequency of copy number of that gene. In addition to that, the CYP9M1 gene, which is also assigned to confer resistance, is also found under selection, but the selection was weak. It is not as strong as the other gene that we have seen on the chromosome 2R. So in generally, the signal of selection across the signal of selection in Anopheles arabiensis in Ethiopia is vary also across sites. As I said, the carboxyl esterase juvenile hormone gene and VGS genes are under selection in the northern Ethiopia, and genes associated with the ubiquitin proteasome systems are only found under selection in Asosa, western Ethiopia. GST2 gene was found under recent positive selection in central Great Rift Valley of Ethiopia, while the CYP6AAP gene cluster was found under strong selection in all cohorts. But we did not observe any difference in signal of selection between seasons. So the signal of selection also vary across season and also across geography. So the question here is, why resistance variants differ across location while the vector control strategy is similar? As I mentioned it in the introduction part, Ethiopia has been implemented the indoor residual spraying and long-lasting insecticidal net intervention, which was applied uniformly across the country's malaria endemic area. But we are detecting variation in insecticide resistance variants. So this can tell us, insecticide resistance is not only driven by the vector control intervention, it is also impacted by environmental and different behavioral factors. So one of the cases could be agricultural pesticide run of agricultural pesticide usage. If we look the evidence on agricultural pesticide usage pattern in the northern part of Ethiopia, farmers, they use pyrethroids and even in some cases DDTs. And also there was also a persistent history, DDT usage history. So these pyrethroid-based and DDT-based interventions might be increased the frequency of target site resistance at the VGSC gene. When we look on the western part of Ethiopia, also, it is the area where large and small scale farmings are involved. And in that area, farmers, they use broad spectrum pesticides, different pesticides. And in the case of implementation of broad spectrum pesticides, the copy number variation which involved in metabolic resistance could be increased in frequency, while the target site SNPs might be, declined because of fitness cost. The other is the human behavior. Communities, they use different commercial pesticides for vector control, and this might have been also impacting to the variation in insecticide resistance variants. Vector behavior by itself could also impact the variation in resistance driving. For example, in the northern part where most of the areas are highland and cooler, mosquito may prefer to rest indoor, so they can have a frequent contact with the intervention, indoor-based intervention like the ITNs, and the frequency of the L995F might be increased. But in others where mosquitoes are outdoor-based, that frequency of L995 might be decreased. And of course, this has to be validated because we don't have even a species-specific comparison on their indoor and outdoor based across the country at national level. But we know little that this can also impact on insecticide resistance. So in conclusion, resistance pattern in Ethiopia is very heterogeneous and dynamic. So northern part of Ethiopia is impacted by target site resistance as the VGSC gene, and this potentially can limit pyrethroid-based intervention, pyrethroid-based ITNs. The western part of Ethiopia is impacted by high copy number variation at the genes which confer resistance to organophosphate and also pyrethroids, particularly the CYP6AAP gene and carboxylesterase genes. So this can potentially limit also both ITN and IRS interventions. The central part of Ethiopia is also impacted by metabolic resistance conferring genes. These are the GST2 and CYP9K1. So this can also have an impact on the efficacy of the pyrethroid-based ITN as well as even for the indoor residual spraying intervention. So this whole genome sequence allowed us to detect the novel signal of selection alongside the known markers, so which can highlight the importance of genomic monitoring to inform vector control. And the resistance, we also understood from this resistance in Ethiopia is driven from heterogeneous factors, vector control and agricultural pesticides and the vector behavior and community behavior. This complex interplay among the different factors might be driving the heterogeneous or variable frequency of insecticide resistance variants. So from this finding, it would be good, it is good to integrate genomic surveillance into the routine entomological monitoring. If we integrate genomic surveillance into the routine phenotypic entomological monitoring or insecticide resistance monitoring, then we know easily where to deploy our intervention, which gene is, which gene in which area is driving the resistance, so where to deploy our interventions appropriately and how to manage the insecticide resistance also appropriately. So it is a good integrating of the genomic surveillance into the entomological monitoring system. The other is it is also good to investigate the real impact of agricultural pesticide, human and vector behavior on driving resistance in malaria vector. And also it is good it is good to validate the lesser known insecticide resistance genes, which we assume potentially they can confer resistance. So this has to be done for the next. Lastly, but not least, I'm very much grateful to all supervisors, mentors, and co-authors. So I'm very much grateful. Professor Lamu Golasa is my advisor and the contributor for MalariaGEN from Addis Ababa University. Professor Hapte also my advisor. And special thanks to Dr. Kelly, who continuously mentoring us and supporting us, and who is still helping us in all aspects of this project or this our study. Dr. Chris, Dr. Arista, Anastasia, and all colleagues are also acknowledged for their valuable contributions to support us. And also I would like to thank all institutions involved for the successful achievement of our study. Thank you very much for your attention. Over to you, Dr. Kelly.
Thank you. Thank you for such a great presentation. I thought it was extremely well presented and raised lots of very interesting questions. Great to see the first whole genome data analysed from Ethiopia for arabiensis. And yeah, raised very interesting insights into how much spatial heterogeneity you can see in insecticide resistance and all the factors that can actually influence that. It's not just a simple relationship with the intervention strategies that are going on in a particular location. I'm going to share your preprint on the chat box. So anyone who wants to actually read the paper, it is available online, so you can fully look through all the results there. We do have time now for some questions, so I'm going to open the floor to others to comment, and please feel free to either put your question in the Q&A box or in the chat box. If you raise your hand, I think I may be able to unmute you as well. So any questions you have, please feel free to ask those now. While we wait, maybe I will ask one of my own questions. So you saw some changes in frequencies of insecticide resistance markers across the seasons, which I thought was really interesting. Do you know of any other studies that have shown seasonality? And given that you've seen this variation, what do you think the implications are for vector control?
Yeah, thank you. That's a very interesting question. So far, we don't have seasonal data to look into the insecticide resistance. Even we don't have data on the genomic markers to look into seasonal. This is the first study that tried to set baseline for seasonal variation and geographical variation as well. But what we can say in here is, in some areas, we know that during major transmission season, the frequency of insecticide resistance variants increase because intervention is also increased by that time. The breeding habitats also increased by that time, and mosquito populations increase. Then with the high exposure to the intervention, the frequency of variants which confer insecticide resistance increase as well. During minor transmission season, in some areas, there are agricultural activities, where farmers, they use groundwater by just setting canals, and those canals, they create breeding sites. And also those in that area community, they use intervention to protect themselves from malaria. So in that specific area, the resistance variants might be also increased even during the minor transmission season. That could be the things that we also saw, that we also detected variation across seasons as well. That is one another thing. So what it can tell us is we need to tailor our vector control strategy. It has to be based on local evidence. It shouldn't be just by saying deploy ITN nationally or something like that. It should be based on local evidence. Thank you.
Great, thanks, Araya. It sounds like it could be really hard to untangle agricultural practices from malaria intervention strategies. Do you have any thoughts on how that could potentially be addressed? Is there any way we could sort of look into the interaction of agriculture and the influence of that on insecticide resistance?
Pardon me, please. Sorry.
Oh, I was just wondering, it seems like, there are seasonal differences in both agricultural practices and the intervention strategies that are applied. So it's quite hard to untangle the two factors. Do you have any thoughts on how we could determine how agriculture is influencing insecticide resistance?
Yeah, that is one of the main challenges. In Ethiopia, there is no integration between agricultural activity and the public health aspect. On the agricultural activity, farmers, they use different insecticides formally and informally. Then, mosquito larvae are already exposed to different chemicals which have the same mode of action with the insecticides that we use them for vector control. So what is required here is, integrated management is, important. We have to document all pesticides that are implemented for agriculture, and then we have to look to our insecticides which we implemented for vector control. then by checking that and by conducting routine monitoring, especially by using genomics, we can predict which vector control tool insecticide is going to be threatened by insecticide resistance or not. We can untangle with that, I can say.
Thank you. So I see we have another question in the Q&A box. So Joel is asking, have you run any bioassays in these areas alongside the whole genome sequencing? I'm wondering whether you have observed any phenotypic patterns that can help explain the marked SNP patterns and their potential operational impact.
Okay, that's a very interesting question. So for the samples, we did whole genome sequencing. We did not phenotypic them, but we have conducted phenotypic assay at similar time on that areas, and we found the northern part of Ethiopia is, the vectors are highly resistant to deltamethrin, and also unexpectedly we found susceptible. The vectors are susceptible to deltamethrin in the central Ethiopia, where we found low frequency of L995F. But across the whole country, for all the sample we whole genome sequenced, we did not conduct phenotypic assay.
Fantastic. Thank you. So just a comment also from Mercy. She said, thank you very much for your presentation. I really enjoyed it. I wonder what the possibility is of gene flow across the locations. What would happen if the mutations flow? How would it affect the interventions you already highlighted?
Thank you very much. Well, this is a very interesting question. So regarding the gene flow, for example, if we look the Central Great Rift Valley of Ethiopia, where we found low frequency of the CYP99, we found high frequency of GST2 and CYP9K1, but in other side we found CYP6A gene high frequency. We did not observe them in the Central Great Rift Valley. So possibly, the Rift Valleys, but that high terrains which serve as geographical barriers might be also serving as a gene flow barrier. That could be one of the possibility. And we have also another work that we showed whether population structure and genome connectivity has impacted on the insecticide resistance variants. We did not observe on that. There is population structuring within Ethiopia and even across East Africa, but the insecticide resistance conferring genes are still, we found them across the whole country. So gene flow is there as it is, and what it is requires is, a transboundary integration or collaboration matters in this case to maintain the vector control strategies, to increase their lifespan. That means.
Great, thank you for that response. That's really interesting to hear that also, there's an added level of complexity in Ethiopia because there are geographical barriers as well, just to add to the complex picture of insecticide resistance across the country. Just see if there's any other questions. Does anyone else want to ask anything else? Araya, do you have any last comments on, insights or, observations from the data that you think are really important to take into account when considering vector control, given the findings that you have?
Yeah, so as I tried to mention before, this is the first whole genome sequence data across multiple sites and across seasons. This is a very good baseline data that the country should explore to, plan for the next, what has to be done for the next. Because as we see the results, the Western part is, highly impacted by metabolic resistance conferring genes. That means Ethiopia is implementing indoor residual spraying using clothianidin now. So even clothianidin, it can be impacted by the high copy number variation because metabolic resistance. There are evidence also metabolic resistance can also, limit the efficacy of indoor residual spraying, even for the new generation insecticides. So these data are very, very important, to set plan and to go further how to implement our interventions.
Thank you. We've just got another question in the chat, but you may have already just answered it, but I'll read it out anyway, and maybe there's something to add. It's thank you, Araya, for the nice presentation. What are the implications of regional and seasonal resistance variation for malaria control programs in Ethiopia? And do you recommend region-specific interventions?
Yeah, thank you very much. So what I recommend is based on this finding, for example, in the area where we found high metabolic resistance comparing genes, it would be better, for example, to implement the dual AI nets like the chlorophenapir based, because chlorophenapir has another mode of action which affects the mitochondria of the vector, which is pro-insecticide, which is also activated, by the presence of high concentration of CP450. And also in the area where we found high frequency of L995F target site resistance, we could implement the indoor residual spraying with clothianidin, for example. So such kind of tailored interventions are important, not only for Ethiopia, but for other countries as well, depending on their evidence.
Thank you. So I think we'll call it a day at this point, but thank you so much again for presenting your data. It was so interesting. You have many compliments in the chat box with everyone thinking that it was such a great presentation. So that just leaves me to promote the next journal club, which is on the 9th of June, and Harun is going to be presenting. The title will be The Role of Genomic Surveillance in Malaria Elimination Strategies and Plans in Africa. So if you are able to join, it would be wonderful if you could come along to that journal club, and I hope to see you there. Thanks, Araya, again. Bye.
Thank you so much again for having me.
Thank you.